bad impacts of broad spectrum chemical pesticides are they generate a lot of resilience in the non-target pest. Particles should be used. What are the safe criteria for nanoparticles to be used for different crops? Because the absorption pattern of crops is very different, and then the consumption pattern is different. So we thought that you just can't have one size fit all. But then we realized उस one size fit all के लिए भी कोई standard नहीं है. So we published this paper, and we raised the urge that we should see about the prospects and challenges in India. And based on that paper, Department of Biotechnology, they called us and they said that we are going to form a committee to see how to form the guidelines for this. Now, so we developed the nanoparticles, but then we chose something because, as I said, I wanted to blend the most traditional rural and low-cost resource available to the modern high-tech nanotechnology in tea production. So we decided to, we decided to make nanoparticles, but then we chose cow dung and cow urine as our base material. The reason was we always thought that this is a good soil conditioner. It was available to the farmer at the low price. So we first conducted a brains. Unilever pitched in, and Tata's also pitched in, and they said that at that time Greenpeace has raised a huge concern about tea having DDTs and pesticides and all. So they said, okay, fine, let us fund a study, and we did this study. It was first we started with a cohort study, and then we did it a real big survey. So we found that 1031 species of arthropods are associated with tea, because tea is a monoculture, and whenever you have monoculture, you have colonies of pests in the comfort zone continuously enjoying it. Then tea alone is one such crop. Every part of tea is consumed by pest, every part of the bush. And the annual loss for a dollar crop is 11 to 55%. Now, tea is not a cereal crop in our country. It is grown in very selective areas. So 11% to 55%, I mean, like, just look at the range, is fairly high. And particularly when your yield is less and your income is more, this really becomes a very impactful thing. And there has been very heavy use of synthetic chemicals for it. So as I just said, every part, starting from the bush to the root, every part is continuously used by some or the other pest. So one size of any agrochemical was anyways not fitting in. So we spoke to a couple of people who were, particularly there is a place in Darjeeling called Makai Badi. And I said, I only drank Makai Badi tea, so it tasted so different. So I traveled to Makai Badi, and then I asked the people, what do you do which nobody else is doing? So they said, you can't spray something for which, so many pests can die. There is no one such molecule. But what you can do is as a practice. And what that practice would be? That practice would be fumigation. So we just heard about homotherapy on the ground floor. So that homotherapy was like, it did not allow the pests to settle. There were certain resins they walked through in the field. It did not allow the pest to settle and then damage. So it was very quick, and it did not even destroy the crop, it did not even give the residual effect, and it was uncomfortable situation to all the pests which were around. It was not entering, it was not systemic, it was not entering into any of the pests. So we published a paper for that. And before we actually jumped on to develop nanoparticles, we thought that possibly color attraction would do something. So we did this yellow sticky traps on the tree. These are like yellow sticky cards in cereal crops we have. And tea is like one single farm, one single tea garden is that big, so it was not possible. So we just covered the tree, the shade trees. And then this was a closer view, wherein you can see here the damage. These are the commercial assets. Tea only leaves are sold, and they are severely impacted. You don't see it in the bigger field, but when individual leaves are plucked, they are affected. So we classified them into different, like what are the major ones, at what stage do they impact, what is the loss of each one of them,
and people have some questions.
What is the loss of each one of them. And we also specifically narrowed down on one pest which did not affect the top layer, that is the commercial layer, but it affected the bottom layer. Now, what was our interest in something which is affecting the non-commercial layer? It was making the plant weak and more vulnerable, more susceptible, because that is called the mother bush. It consumes the mother bush, no photosynthesis, no strength in the plant. So we chose red spider mite, and from there we chose cow dung and cow urine as a base material. We went to the British Ayurveda and to Srimaharshi, and in which it is very clearly written about Gomai Vasate Lakshmi, which means Lakshmi, goddess Lakshmi resides in cow dung, and we did not know what it meant. So we thought, okay, does it have gold? No, it did not. Ornaments भी नहीं आ रहे थे बाहर उसमें से. We tried it. We really did it. We not search for ornaments, but we tried to dry it. We tried to make liquid slurry out of it, but we just didn't know Lakshmi kahan hai. We just didn't know. Then we thought possibly we should do the profiling of it. So more than 10% profiling we could do. There are so many microbes, and these microbes, some of them have excellent soil conditioning, pest repellent properties. So we thought possibly, in a colloquial term, we'll take it as this is Lakshmi. So we started with cow dung, assuming it to be the gold mine of microbes, and then we realized it also has insecticidal properties. Then cow urine. We all know that Agriculture University of Junagadh, they did a project and they extracted gold salt, Swarnshar. They extracted from cow urine, and when I read that report and I met the scientist who did it, I was at least concerned, okay, that Lakshmi part comes here. No. I mean, I did not read the Vedic, so my misinterpretation, kindly overlook and forgive. But as a layman, if I say I read something and then I don't find any evidence, then I don't even find somebody to help me to interpret. So this is a disconnect which we have. So there was gold salt. I mean, Swarnshar was actually found, and this is a published paper. So and this also had a lot of insecticidal properties. So we prepared and characterized the infected leaves here. Then we also made a cow dung slurry. It was like huge, and the tea garden owners were like, tea is something which people drink for flavor. And imagine cup of tea has cow dung or cow urine flavor. So he wanted to make sure. I mean, his only concern was, you will give me a smell test that this tea does not have cow urine smell. But when we were making cow urine nanoparticles, we did not have that cow urine smell at all. Even in the cow urine liquid, we did not have that smell. So I said, now how do I do that smell test for you? Because it does not get retained in the dried made tea. It is a fermentation process. Then they also said, cow dung ka taste nahi hona chahiye. It is something, if I tell you, इसमें Japanese meal का taste नहीं होना चाहिए, and you have never tasted Japanese meal, how would you really say? I never tasted cow dung, so I just don't know उसमें cow dung का taste है या नहीं. So these were the questions which came to me like bolt from the blue. ऐसे taste तो कैसे होगा? Cow dung से nanoparticle बन गए नहीं, cow dung का taste चाय में नहीं और cow urine का smell चाय में नहीं. So these were the two conditions he made me sign, and we said okay fine. So we did this test in 4 tea estates, and then we synthesized zinc and iron. We took it because they were even deficient in the nutrient, as I said before. So zinc and iron were they usually make the plant resilient, and this was the cow dung extract, and from here we made the nanoparticles, and then we did the analytical studies. So we did spectroscopic studies at 30, 40, 50 and 60 degrees, and we found that at 50 degrees we were able to see some very weak peaks. And then we did it for effect of salt, when we want to make it as a salt. So we also see, is there any impact on salt? Then we did SEM, EDX and FTIR also. And then we made a control. The control was made by propagandite, which was the common acaricide, because it is a mite, a red spider mite. It is killed by acaricides. So the common acaricide was propagandite chemical. So we made the control for that. We made a positive control as neem, because neem was green. and it was not cow dung and it was not cow urine, so the tea owner was okay, fine with it. So we found that the most effective one was zinc oxide nanoparticle at 1000 ppm, and it was 1000 ppm again on eggs. Like we just did for two methods. One was leaf dip method and another was spray method. So we dipped the leaf in the solution and then we sprayed it over the solution, and we found that is absorption making a difference because it was nanoparticle, it was getting absorbed anyways. So we found that in both the cases, our 1000 ppm was much better than control, and it was even better than the most popularized neem bioproduct. And then we did it on eggs. The previous one was on adults, and this is what we did it on eggs, and we again found the same result, that the cow dung was giving much better protection. We did repellency test, that does it cause repellency or ovipositional deterrence. So the concentration ppm, and we found that 1000 ppm was actually doing good justice. It had no phytotoxic effect. Nobody died. I was the first one to taste it. I am still alive. Plant did not die. So no phytotoxic effect was there. No visible injury was there on the tip, on the leaf tip or the commercial part of it. And there was no wilting, necrosis, or all the signs which we call as ill plant health. Organoleptic test also we did. That means the taste and smell and aromatic test, and it was found fine. The tea garden owner was much relaxed, much more than me. Now this is what we did with cow urine. When we made nanoparticles, this is what we did. And then again the same test. We did the effect of temperature and the effect of salt concentration. And we did SEM, EDX and FTIR for those. And then we did the efficacy test. So here also our control was much more in leaf dip and spray method as contrast to the neem and the control one. And this is on fecundity and egg laying capacity and then the eggs. So we found that it was doing very well in eggs, thereby breaking the life cycle of the red spider mite itself. There was again no phytotoxic effect of cow urine also. I'm still alive. And there was no visible injury. And when we did the tainting test, the score was 6.5 to 7, which is considered to be a very, very safe score. So advantages were, it did address the major challenges of emerging contaminants. Residue in the leaf, we were not able to detect it. Major dilution we did, we were not able to detect. Bioaccumulation was not there. Non-target species destruction, we did the entire experiment on spiders, because spider is a friendly insect in the tea gardens, but it did not kill spiders. So it is not targeting the non-target pest. It was easy and it was less costly in contrast to the chemical ones. So the future scopes are to study the better effectiveness, bioavailability and environmental sustainability, and good for commercial scale up. Now there are barriers. When I say that this is a technology and it needs to be developed and it can be upscaled, so I sum up all the barriers in 3 ways. The whole property of cow dung is very much animal centric. So if the animal is not well, that means if the cattle is not well, or the feed is wrong, or if the real-time health data is not collected. So we do have cattle sensors on the ears they plug in. But first, it does not give correct data. Second, the data is huge. So interpretation of data is again a problem. And then the third one is when somebody is having, gaushalas are usually sourced for large scale production of nanoparticles. So gaushala to the industry is not mapped on blockchain. So these are the 3 B's which are the barriers. And 4 R's to make any green revolution technology more sustainable is robustness. It should be very robust. It should be recovered, and reorientation and reorganization. So we tried to achieve all 4 in our experiment, and that was it. The tea is safer than before, and I'm still alive, so nobody died drinking that tea. Yeah, and I still drink tea. So I would say that we can innovate with technology more. There is so much more work needs to be done in this spectrum, in this segment. There is always a better way to do things. Possibly the same experiment if somebody else does, but does it better, even better. So these are the coordinates if somebody would like to be in touch. Thank you so much.
Thank you, ma'am.
Sir, at least by 5% when we finished up with the product, 5%. Now it has been 7 years we have completed it because we took 3 areas, Kanda, we took Northeast and then we took the Southern Delhi hills. So
quality, taste is
quality is much better. Quality is much better. The tea leaves are softer and then the aroma retention is higher. Now I have no reason to explain the pathway of aroma retention. I'm still doing it, but this is an outcome.
But the price of the tea also might have gone up.
No, it did not, because cow dung is a very rural resource. So had it been like, I would have said use gold cow dung or gold nanoparticle or something.
Because of the increase in the aroma and yield, the farmer will be getting much more.
Sir, actually, tea is sold in auctions. So auction is always a consolidated price. It is not yield based.
Okay, any questions?
I have a comment only.
Yes, please.
Basically, with my interest in the biodiversity part of it, I do follow the natural farming processes where actually, the cow dung and cow urine are the main inputs. Essentially, that natural farming system focuses more on the improvement of the soil ecosystem, microbial ecosystem. But in tea, it is not possible. It's only possible when you are doing an annual or biennial crop, not for the perennial crop. There actually I observed that most of the times the advocates of natural farming speak about using only the cow urine or cow dung of the desi, the local breeds, not the hybrids, that are used essentially for the higher productivity, higher milk yield, etc. Because actually they have a logic also. They say the microbiome, gut microbiome is far different within the desi cows as compared to them. Perhaps my only thought is you have to consider that aspect and then do some understanding, have little understanding. Perhaps in terms of the pesticidal efficacy also, the microbiome characters, it's too complex, one cannot study the entire, and I do understand about it. However, but at least whether there is any functional difference in terms of the pesticidal activity.
Sir, thank you so much. This is a very important aspect because we took cow dung consistently from Gaushala, which has a mixed breed. So if we actually segregate it and then see, because I said in one of the barriers where the cattle is given punched with a sensor, the data which comes largely comes on health aspect, like the cow is not pregnant, the cow is healthy, it is not having that foot and mouth disease, but it does not speak about the microbes and the breed. So possibly that is an important thing I am missing out. And
if it is a Gaushala, it is a native cow.
It is a native cow. Okay.
One more important point to add to Sar's comment. You have used this cow dung separately and mixed with water, or how did you use them?
Yeah, we made a slurry of it.
Water and normal water or tap water?
Normal.
Then urine also you mixed separately.
Urine was already diluted. We did serial dilution when you wanted to do the microbial.
What was the proportion of water and cow dung?
We did serial dilution, so it was 10, then 1 and 9, 2 and 8, 3 and 7, 6 and 4, 7 and 3, like this. So when we were able to draw the pure culture out of it, then we kept on diluting it.
In all respects, I compliment you for an excellent oration. And the innovation that has gone into it. Thank you.
Thank you so much.
Dr. Vrindavan worked in Jobber for many
Worked in
years.
Dabur earlier, but my passion is in biodiversity.
But traditional farming always no cow dung is being used in farming.
It's a good moisture also it used to solve.
Why not the slurry of cow dung with cow urine?
Jivamrutam, I have not finished my comment.
Sorry,
So if you use your Jivamrutam,
yes.
what is being suggested by all our organic farmers?
Right.
Like use cow dung, desi cow urine, within 24 hours only you have to use. Maybe a liter with 1 kg of cow dung. Then use some chana powder.
Yeah.
Then gur, jaggery. Mix it, store it for 15 days.
Okay.
Maybe after 3-4 days it starts giving a very good
Balance.
bioavailable smell. Also you will find the different smell. So from that day to 15 days. If you use that, you will not have the pest also on the plants.
So you are suggesting I should make nanoparticles from Jivamritam? This is what it is?
If you are using separately, instead of that, if you use it from Jivamritam,
Right.
it may give you wonders.
I should try this. I will surely.
I am already using it for my terrace garden.
So we can
I'm
actually,
getting
yes, we can actually talk more.
Note down the email address and then contact.
Yes, please, we can.
Dr. Kosraj?
Yes.
One quick clarification. The microbiome, it changes depending on the type.
So it does. It heavily does. Because the cow's stomach
the
is.
desi cow and the.
Yeah, actually, I can only speak of diet because lignocellulose heavy material.
Grass
like
fed cow and
the,
grain fed.
the grain fed cow, it differs. The lignocellulosic material, particularly in the Punjab, Haryana side, where they have so much of rice parali, the rice stubbles, it is very, very dense on cellulose, and the cow's stomach doesn't digest it very comfortably. So then you find a lot of fibers in the cow dung, and then it is difficult to convert them into nanoparticles because they keep on, silica keeps on interfering. And silica is, among nanocontaminants, silica is a huge contaminant. It is very difficult for me to just release out silica. It is difficult for me to make a nanoparticle and does not have silica contaminant in it. It is that.
There are many factors which influence anyway in terms of the application and the effects also. I think let me congratulate Dr. Kavya
Thank you.
for excellent.
It's a wonderful work, man.
Thank you.
It's a very innovative.